Welcome back to the deep dive. Today we're taking on probably the greatest mystery machine in the known universe, the nervous system.
It is the ultimate processing center.
We're diving deep into the architecture that controls every single action, maintains your internal balance, your homeostasis, and, ultimately defines consciousness itself.
And our mission today is to try and cut through that incredible complexity. We're going to trace the nervous system's really surprising evolutionary origins, look at the core machinery, and then
jump all the way up.
All the way to the mathematical frontier. These sophisticated top-down models scientists are now using to try and predict its collective activity.
Okay, so let's start with the fundamental structure. At the highest level, you have the central nervous system, the CNS.
That's the brain and spinal cord, the command center.
Right, the protected core.
Yeah.
And then branching out from that, everywhere, is the peripheral nervous system, or PNS.
The wiring. It's the network relaying all the information back and forth. To really get this, we have to start with the basic building block, the neuron.
And what makes this cell so unique is its vulnerability, isn't it?
It is, and frankly, it's a little terrifying. Unlike most cells in your body, neurons are generally not replaced if they're severely damaged or lost. You get what you get.
So it's a precious resource. Structurally, these cells are just optimized for speed and direction. You have the dendrites.
Like branches.
Yeah, they collect the signal and pull the impulse toward the cell body. And then the long transmitting wire, the axon, that carries it away.
Always away. It's a one-way street. And how fast that signal travels, that's all about insulation.
The myelin sheath.
Exactly. A fatty layer that coats many of the axons. You can think of it like the plastic coating around a copper wire.
It prevents signal leakage.
And dramatically increases the conduction speed.
And that difference in insulation is something we can actually see, right, when we look at the brain.
We can. The unmyelinated tissue, where you have all the cell bodies and connections, that's what we call gray matter. And the myelinated tissue, all those long-distance fiber bundles, that looks lighter. That's the white matter. The system is physically organized for speed.
So the signal travels down the axon and reaches the end. What happens then?
It hits the synapse. That's the tiny gap between neurons. The electrical signal doesn't usually jump that gap.
It triggers something else.
It does. It causes these little packets, synaptic vesicles, to release their chemical cargo, the neurotransmitters, into that gap.
And they just float across and bind to the next cell?
They do. But here's where we hit one of the most surprising points, one that trips a lot of people up.
Okay.
The effect of that message, whether it's excitatory, telling the next cell to fire, or inhibitory, telling it to stand down, it does not depend on the chemical itself.
It depends on the receptor it binds to.
Exactly. The neurotransmitter is just the key. The receptor is the lock that determines what happens when the door opens.
So a chemical like, say, glutamate is famous for being excitatory.
Right, because it typically binds to receptors that cause excitation. And GABA usually does the opposite, causing inhibition.
But a single neurotransmitter could have opposite effects on different cells.
It can. Acetylcholine is a classic example. It can bind to completely different receptors and cause opposite effects. It's an incredibly flexible system.
And there's this other idea, Dale's principle,
which basically says that a neuron will generally release the same neurotransmitters from all of its endings.
But because the target cells have different receptors, that single neuron could be telling one area to fire up and another to calm down, all at the same time.
It's incredibly efficient.
So let's quickly frame the peripheral nervous system. We divide it into the somatic and the autonomic systems.
Right. Somatic is largely your voluntary system, sensory input from your skin, moving your skeletal muscles.
The things you decide to do.
And the autonomic nervous system is, well, it's completely involuntary.
That's all the stuff you don't think about. Glands, internal organs.
Pupil size, everything. And it's governed by this constant tug of war.
Between the sympathetic and the parasympathetic systems.
The accelerator and the brake. Sympathetic is your fight or flight response. Emergency mode. Heart rate goes up, digestion shuts down.
And the parasympathetic is the brake.
It's rest and digest. It slows the heart rate, stimulates digestion, and brings the body back to its baseline. They're always in balance. Now, if we look back evolutionarily, this incredible complexity didn't just appear overnight.
Right.
Tracing the path reveals some profound differences and even some what you might call shocking evolutionary flips.
The story starts way back with sponges.
It does. They are the absolute base level. They have no neurons, no synapses, nothing like that.
But they have the genes for them.
They have the genetic blueprints. The homologs for synaptic genes are there. They just haven't been assembled into a system yet. They communicate with these slow calcium waves to do simple things like contracting their whole body.
So a step up from that would be jellyfish.
Jellyfish and comb jellies, yeah, they have the first nervous systems, but they're completely decentralized. Just these diffuse nerve nets spread across the body. There's no brain, no center of control.
The big leap then happens with the bilaterians.
Most life on Earth. Animals with that approximate left-right symmetry, they developed a centralized nerve cord.
And a gut.
And a gut. And this is where that upside-down concept comes from. It's fascinating.
Unpack that for us.
Okay, so you have protostomes, think insects, worms, mollusks. Their main nerve cord runs along the ventral or bottom side of the body.
Okay.
But in deuterostomes, that's vertebrates, including us, our nerve cord, the spinal cord, runs dorsally along the top.
So the main hypothesis is that an ancestor just flipped over.
Essentially, yes, about 550 million years ago. And it's supported by gene expression patterns. The structure that runs our entire body is, evolutionarily speaking, upside down compared to a fly.
That is genuinely 1L.
It is. And while we operate with this massive interconnected complexity, some simpler animals give us a glimpse into total neural certainty.
You're talking about identified neurons.
Oh, yeah.
Give us the definition of that, because it sounds like a perfect precision-engineered part.
It is. An identified neuron is one that is so unique in its location, its chemistry, its connections, that every single individual of that species has one and only one with those exact properties.
And humans don't have these.
I have none. Our brains are plastic, our connections are shaped by experience, but for some animals, it's all hardwired.
Like the roundworm C. elegans.
The perfect example, its nervous system is completely mapped. Hermaphrodites have exactly 302 neurons. Males, 383.
And every single one is identifiable.
Every one. Their entire neural structure is genetically specified. No learning required.
And in vertebrates, the classic example is the Mothner cell in fish.
Yes, these two gigantic, easily visible neurons in the brainstem. If a Mothner cell fires just one action potential,
Just once.
it's a powerful enough signal to trigger the entire escape response. The fish instantly bends into a C shape and rockets away from a predator.
It sounds like an override button. A true command neuron.
It does, and that's what people called it. But that concept, the command neuron, is a bit more controversial now.
Why is that?
Because context always matters. While the Mothner cell can drive that escape, other studies show that many of these supposed command neurons only work under very specific circumstances.
So the wiring might be genetic, but the performance depends on the situation.
Precisely. The certainty is there, but performance is context dependent.
Okay, that was a perfect transition. We've just talked about systems where we know every single wire 302 cells, 2 Mothner cells. So how do we jump from that to modeling a human brain?
With billions of cells.
Yeah, with non-uniform connections and, frankly, organizational chaos. How do you even start to model that?
You can't, not by tracking every single neuron. That bottom-up approach is computationally impossible.
So you have to abstract.
Exactly. The solution is abstraction, and that's where the math comes in. It's a bit like weather forecasting.
How so?
Well, you don't try to track the position and momentum of every single air molecule. Of course not.
You look at population dynamics, average temperature, mean pressure, wind speed to predict the overall state. Scientists use neural field models, or NFMs, to do the same for the brain.
So you're focusing on average properties of the whole population.
Exactly. Mean firing rates, mean membrane potentials. It keeps the math tractable.
What are the key building blocks for that kind of model?
There are 3 main pieces. First, you have what's called the wave-to-pulse transfer function.
Okay, what does that do?
It's basically a function that translates the average voltage of the cell population into its average output, its firing rate.
So it's like a threshold. If the average excitement level gets this high, the group as a whole starts transmitting signals.
That's a perfect way to think of it. It's usually a sigmoidal or S-shaped function. Second, you have the temporal kernel.
Which sounds like it deals with time.
It does. It captures how a signal decays over time once it arrives at a synapse. A signal isn't instantaneous. It sort of echoes and fades.
So the temporal kernel is the reverb of the signal. Great analogy. And third is the spatial kernel. This one is vital because it shows that not all connections are created equal.
I'm guessing this one tells the model that connection strength decreases with distance.
You've got it. It assigns weight based on physical proximity and connection density, just like a social network.
And this whole approach really took off in the 1970s, didn't it?
It did. The huge breakthrough came from Wilson and Cowan. Their models changed the field because they were the first to explicitly include spatially extended masses of both excitatory and inhibitory neurons.
So they modeled the tension, the push and the...
Exactly, they formalized that tension, which is absolutely fundamental to stable brain activity.
And I imagine since then, the models have only gotten more complex.
Yes, there's been a shift toward more biological realism. Modern models often focus on modeling the mean postsynaptic potentials, or PSPs.
The immediate voltage changes from a signal.
Right. And that lets the models capture different timescales more accurately, like how excitatory signals are usually fast and inhibitory signals can be slower.
And for simpler problems, we have neural mass models, NMMs.
Which are just the field models constrained to a single point in space. You drop the spatial part, they're huge for understanding things like EEG rhythms.
Like the Janssen and Rip model.
A classic NMM. It uses 3 populations: pyramidal cells, excitatory interneurons, inhibitory interneurons, to simulate the electrical activity we measure on the scalp.
And building on that, the Wendling model, with 4 populations, it actually led to a really influential finding about pathology, didn't it?
It did. The Wendling model suggests that a critical reason for the transition into a seizure isn't necessarily a huge spike in excitation,
But a failure of the brakes.
a failure of dendritic inhibition. If the brakes fail, the system just runs away with itself. These models let us test those very specific hypotheses.
So these mathematical models, they aren't just academic. They're fundamentally changing how we understand real-world disorders.
Absolutely.
Let's talk about the system's vulnerabilities. The CNS, brain, and spinal cord is incredibly well protected.
Physically, you have the skull and vertebrae. Internally, you have membranes called the meninges. And crucially, you have the blood-brain barrier.
A chemical shield.
Right. It controls what gets in from the bloodstream. But the downside is, when damage does happen, say to the spinal cord, it's often permanent. Scar tissue prevents nerve fibers from regrowing.
Whereas the PNS, the body's wiring, is more exposed.
Things like carpal tunnel, sciatica,
But it has a better capacity to regenerate.
a much better capacity. It can take months, but it offers a chance at recovery the CNS rarely does.
We've already touched on epilepsy with those storms of abnormal electrical activity.
Right. Then you have autoimmune issues like multiple sclerosis or MS-PO. That's where the body's own immune system attacks the myelin sheath.
It strips the insulation off the wires.
And that just devastates signal transmission. Then you have the degenerative diseases. Parkinson's disease is the loss of dopamine-producing neurons in the midbrain.
Which leads to those characteristic motor symptoms, the tremors.
And Alzheimer's, which is just this catastrophic, widespread degeneration leading to loss of memory and cognitive function.
So how are these models helping here?
They're focused on 2 main things. First is rhythmogenesis, understanding how the brain generates its normal waves, your alpha, beta, delta waves.
And the second?
State transitions. How does a normal rhythm suddenly flip into a pathological one, like a seizure, or a necessary one, like falling asleep?
And this is the frontier, right, predicting that shift. I've heard about this idea of critical slowing.
It's a very exciting concept. Critical slowing is a phenomenon you see in complex systems right before they undergo a dramatic shift.
Okay.
As the system approaches that tipping point, it gets sluggish. The speed at which it recovers from any small disturbance slows down dramatically.
So if you can detect that slowing down in real-time EEG data using these models, it could be an early warning that a seizure is imminent.
That's the hope. A warning system measured in minutes or even seconds. But this brings us to the single biggest challenge in the entire field.
The sheer number of variables.
Exactly. Despite how elegant these equations are, they have far too many unconstrained parameters. We have to estimate connection strengths and time constants we just can't measure directly in a living human.
So you risk overfitting the model.
You do. The more knobs you have to turn, the easier it is to make your model look like the data, even if the underlying assumptions are wrong.
There's a famous quote about that, isn't there?
There is. It's often attributed to the mathematician John von Neumann. With 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk.
It's a perfect caution. A reminder of the balancing act between mathematical precision and true biological insight.
It is a phenomenal journey, though. We've gone from the simple calcium waves in a sponge through that bizarre evolutionary flip all the way to differential equations trying to forecast an epileptic event.
The big takeaway for me is that the nervous system is this incredible hierarchical processing machine. It's designed to turn raw sensory data into a useful map of the world.
That's it. Your retina only sees scattered points of light and color. But your nervous system instantly transforms that into a stable, recognizable neural representation of an object, complete with memories and actions attached.
So what does this all mean for you, the listener?
If the ultimate function of this system is linking what you perceive with what you do, consider this provocative final thought: the mirror neuron system.
Cells that fire both when we act and when we just watch someone else do the same thing.
They might be the crucial physiological bridge between perception and action. So, how profoundly does this system influence your ability to imitate, to learn new skills, and maybe even to develop that critical human faculty we call theory of mind, the ability to understand the intentions of others?